Welcome to the session of in fifth semester BSc Zoology core course paper, where we are going to discuss on evidences of organic evolution. And this presentation brings to you the physiological and biochemical evidences uh, of organic evolution. And there are quite a few examples that can actually reflect or light on the organic evolution. The first one being the chemical composition of protoplasm. The body of all living things, it is made up of a common substance called protoplasm, and even each cell consists of a small mass of protoplasm. So the qualitative and the quantitative composition of the protoplasm remains the same, or show a striking similarity from the very lower unicellular group of organisms to the highest advanced group of organisms. The chief elements constituting the composition of protoplasm includes carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, etc. And these elements are uh, incorporated into organic and inorganic compounds of protoplasm. For example, carbohydrates, lipids, proteins, nucleic acids, water, and different minerals. So these elements and compounds are common to all the cells of different types of organisms, and this indicate that there is a common ancestry for all of them. Regarding the DNA composition, yeah, the chromosome chemistry, the similarity. Chromosomes and genes form the physical basis of heredity, and they have the same chemical composition in all the animals. Each, this one is made up of, chromosome is made up of proteins and nucleic acids. Nucleic acids could be DNA and or RNA. The structure of these nucleic acids from viruses uh, through different groups, kingdoms to animals, it is just the same. And the nucleic acid, it is made up of five basic chemical elements, that is carbon, hydrogen, oxygen, nitrogen, and phosphorus. And these elements are organized into tiny units called nucleotides. So we can see here there are different group of chemical studies that can help in providing evidences for organic evolution, and this is actually the structure of DNA. And we can see that the nucleic acid, or the nucleic acid, the DNA or the RNA, it is composed of smaller units, or we can say that monomeric units, the nucleotides. Okay, so it is actually polynucleotide chain that give rise to the nucleic acid. Now, DNA is a long chain of nucleotides, and each of the chain being made up of nitrogen. Each nucleotide is made up of nitrogenous base, a pentose sugar, that is deoxyribose, and a phosphate compound. So these three, nitrogenous base, pentose sugar, and a phosphoric acid or a phosphate compound, come together to form the nucleotide. Okay. Now, the DNA, it is double-stranded and appears like a twisted ladder. And genetic information for all the life activities in whichever be the organism, it is coded in the arrangement of nitrogenous bases between the sugar and phosphate molecules. So these things you are already familiar with, which you have already studied in your previous classes. Um, the similarity between the DNAs of two species was studied by Hoyer and co-workers in 1964. And by suitable methods, what they did is they melted down the DNA strand, double-stranded DNA, and what happened? It got separated into two single strands. Okay, and this single-stranded DNA of one species was broken into smaller fragments, and these fragments were brought into contact with the single-stranded DNA of another species. Okay, the DNA fragments of species 1 were found to pair with only those DNA fragments of species 2 uh, having complementary base sequences to form a double helix. So complementary base sequence, in the sense you are already familiar with the fact that the adenine always base pairs with thymine and guanine with cytosine. That is complementary base pairing. So based upon the complementary base sequences, the DNA strand, the part of the DNA strand of species 1 will base pair with the part of the DNA strand with species 2. But the fragments that were not complementary to any regions of the species 2, it will remain uncombined. Okay, so thus it is assumed that the greater the degree of pairing between the DNA strands of species 1 and species 2, the closer the similarity between the genetic constituents of the two species. So, how far or how much of the what do you call the DNA is base paired, it actually indicate how closer the two species are. So, this particular experiment was conducted by Hoyer and his co-workers, and the DNA similarities can be used to find out the phylogenetic relationship. So, if the two species, the DNA strands of two species are so closely base paired, or the uh, like a maximum amount of DNA is getting base paired with the DNA of the species 2, obviously it indicates it is having a closer relationship, and so phylogenetically it is having a very recent common ancestor. So the DNA of chimpanzee and gibbon, for example, shows greater similarity to human DNA, while hedgehog and chicken, they show least similarity, because the the recent common ancestor, obviously humans, the gibbons and chimpanzees, that is the primates, they have a very recent common ancestor, while the ancestor that have given rise to the chicken, hedgehog and the primates, it have been very early, okay, in the geological time scale. Next, we have the digestive enzymes. Certain digestive enzymes have similar or show similar structure and physiological function in different group of animals. For example, trypsin. You're already familiar. Trypsin is a enzyme that helps in protein digestion. It's a protease. Okay, so this is a protein digesting enzyme found in all animals from very lower group to humans. Similarly, enzyme amylase acts on starch and it is present from sponges to mammals. So this similarity of enzymes, it indicates that there is a common ancestry for all these animals. Then hormonal similarity, if we study, some hormones produce similar reactions in widely different animals when injected. When the hormone is injected to the widely different animals, we can see that these animals show similar reactions. For example, hormones extracted from thyroid gland of cattle can be effectively used in the treatment of human thyroid deficiencies. And this hormone is also essential. The thyroid hormone is also essential for the proper metamorphosis of frogs. If a tadpole's thyroid gland is surgically removed, it will not metamorphose into frog. But if thyroid hormone extracted from cattle or sheep is injected into such a tadpole, metamorphosis will occur. So here we can see that the thyroid hormone from the sheep or the cattle can bring about the same effect as that of the thyroid hormone in a tadpole. Okay. Similarly, there is yet another example of the hormone giving rise to similar reactions in diverse group of organisms, and that is the melanophore expanding hormone. Okay. The melanophore expanding hormone of amphibians causes pigmental cells of the skin to expand, thus darkening the color of the animals. It has no effect in mammals. Yet, if the same hormone is extracted from mammals and supplied to amphibians, it is as effective as the amphibian hormone itself. So here we can see that the one is like its presence in mammals. In mammals, it is actually vestigial, because in mammals it has no effect. Okay, but it is vestigial, it is present, and this can be used in the amphibians. So this can be analyzed only on the basis of descent from an ancestor, okay, single common ancestor, to which the hormone was useful. So the melanophore expanding hormone was present in the ancestor from whom the what do you call the mammals as well as the amphibians had evolved into. Okay. Next we have is regarding the phosphagens. The word itself says phosphagen, phosphate generating, isn't it? So here it is with regard to the ATP generation, right? So phosphagens, it is actually the energy-rich compound in muscles, providing phosphate group for ATP synthesis. So it plays a key role in muscle contraction and are the sources of energy for the resynthesis of ATP from ADP once the ATP are broken down. So when we phylogenetically speaking, the animal kingdom may be divided into two main stock, the chordate and the echinoderm, and the second, the annelid mollusks arthropod. So these two stocks have diverged from a common ancestor millions of years ago, and there is not much morphological evidence with regard to this. However, it is actually given considerable support from the distribution of phosphagens. Okay, so there are two types of phosphagens, phosphocreatine and phosphoarginine. Chemical analysis of muscles have shown that phosphocreatine is found exclusively among the chordates and echinoderms, and phosphoarginine in the other stock or most other phyla. So, in other words, distribution of the same phosphagen occurs in phylogenetically related groups, right? So, phosphoarginine in present in the second ancestral, that is what do you call stock, for example, annelids, mollusks, and arthropods, and the chordates and echinoderms possessing the phosphocreatine shows these two groups have evolved from the same ancestral stock. Okay. Now, the next, the biochemical evidence, it is with regard to the serological tests, whatever is provided. So, it is by way of comparing blood proteins of different animals. So, demonstrating evolutionary affinities to compare the blood proteins by way of comparing blood proteins of different animals. So phylogenetically similar species have similar blood proteins. So closeness on on this basis can be demonstrated by the serological tests. Okay, so this is one of the experiments. If a foreign protein material is introduced into the body of a vertebrate, the recipient produces specific antibodies to the foreign protein material. The substance, this foreign protein material is hence known as antigen because it has stimulated the production of antibodies. Okay, so if human serum is injected into the body of a rabbit, okay, so the human serum is injected into the body of a rabbit, so the rabbit responds by producing antibodies against the antigens in the human serum. The serum containing such antibodies, it is known as antiserum. Okay, the if this antiserum is extracted from the rabbit, is mixed with the human serum, you can see here the rabbit, human serum is introduced into the rabbit. The rabbit produces antibodies. The rabbit serum is collected, and this rabbit serum will be containing antibodies against the human serum, isn't it? So this serum is known as the antiserum, which contains antibodies. Now this antiserum is mixed with human serum. So what happens over here is there is an antigen antibody reaction resulting in the formation of a white precipitate. Okay, and hence this test is otherwise known as precipitin test. Okay, so this is the precipitate is formed. The amount of precipitation gives an indication of the degree of similarity between the different sera. So how much of the precipitate is formed? Okay, that indicates the closeness of the two groups. Okay, now the greater the amount of precipitation, the closer is the serum of that of the human. So here you can see it, the precipitate is formed. Okay, and obviously it is because of the human serum, it shows maximum precipitation. Coming to the chimpanzee serum, so the rabbit's anti-serum was mixed with chimpanzee serum. So what happens over here? If the rabbit's antiserum containing the same antibody is mixed with the serum of chimpanzee in another test tube, a precipitation is formed again, showing that proteins in chimpanzee serum are chemically similar to the proteins in the human serum. Okay, if the same antiserum is mixed with the blood of the baboon, uh, here you can see the precipitate is less, isn't it? It shows that the serum of baboon is less similar to the human serum. Okay, when the